we can get started. So welcome to you all to this full series of the worldwide neuro seminars on the neurobiology of addiction. We started this a while back. We have now had a break for the summer vacation, and we're going to have another 7 talks until Christmas. So on average every other week. They're always run at 6 p.m. Central European Time, noon Eastern Standard Time, except on November 4th, where because of the difference in the switching from the daylight saving time, it's going to be 11 a.m. Okay, so now, with any further ado, it's my great pleasure to introduce Cristina Miliano, who will introduce today's speaker.
Thank you. It is my great pleasure to introduce Dr. Brigitte Kieffer today. She graduated from the University of Strasbourg, where she later became a professor, and she developed her main research activity in Strasbourg in 2001 and directed the institute from 2012 and 13. She spent the last years at the Douglas Mental Health Institute in Montreal, Canada, but she's now back in Strasbourg, France. She is well known in the field for isolating the delta opioid receptor gene back in 1992, and since then she continued to study the opioid system, bringing major advances in pain, addiction, and mood disorders research, as well as in the area of molecular pharmacology and G-protein coupled receptors. During her career, she has been awarded multiple times. She has hundreds of publications in top journals and almost 27,000 citations. So she definitely represents a role model for all women in STEM, and we're very excited to have her today. So without any further ado, I'll give the word to Dr. Kieffer.
Thank you very much, Christina. Thank you for the invitation. Thank you for the initiative of Worldwide Neuro, because I think this truly helped us, among other initiatives, keep our community alive and connected during this adverse time. And I must say, it's pretty adverse to talk to a computer, but I'm happy to see your face. So at least I have one face, or 2 faces with Christian. So I'm going to share my screen here. Is this all right?
Perfect.
Okay. So let me start this way. Opium relieves pain and produces euphoria. So we'll have a little introduction on that. Opium was used in centuries, as you know, and millennium, actually, because this extraordinary substance, in fact, kills any type of pain, physical pain, mental pain, social pain, whichever pain you want to think about. The active ingredient, do you see my arrow? Yeah. The active ingredient of opium was isolated a long time ago, 1805, and this is morphine. And it was clear that morphine, I'm just pushing the little images here, that morphine indeed is able to kill pain, but is also an addictive substance. So from there, chemists actively synthesize, and there's thousands of opiate compounds in the world, and to try and separate the 2 effects. Come on. I'm blocked.
So maybe sometimes you can just do it with your space bar. Is that going to advance it?
Oh. No.
On the bottom, you have the arrows, maybe, yes.
Yeah, that works. Okay. So several years after, the first non-addictive opioid drug, a simple deacylated morphine molecule, was put on the market by Bayer as the first non-addictive opioid to kill pain. And it seems, as I said, the problem was solved. But of course, this molecule, which was called heroin, didn't solve the problem at all. And we know that, in fact, opium and opiates, or opioid drugs, are in the middle of societal challenges since many years. since, for example, the Opium Wars in China, where the British trading policies flooded China with opium, devastating an entire economy, to heroin prohibition in 1920, to until today, where the overprescription of opioids to treat pain has got out of control, leading to, let's say, dependence, opioid dependence, and transition to heroin and fentanyl use, and death by overdose. So you've seen all this type of curves. All of you have seen this. In the meantime, the development of, well, we have developed an extraordinary neurotransmitter system. I think I need to, sorry. Yeah, I have a little problem here. It's okay. So we have discovered an extraordinary neuromodulatory system and modern, let's say, structural biology, pharmacology, and neurobiology or neuroscience has provided incredible tools to study the system. So let's go there. First, I present you the component of the opioid system very quickly. Opioid binding sites were discovered in 1973, followed by the discovery of peptides that normally interact with the receptors. And these were methanol and enkephalin, the first discovered in 1975. A few years later, 3 genes encoding those peptides were identified. You know their names. They encode large precursor protein that are later cleaved in smaller peptides. And you see the larger one, beta endorphin, the smaller one, enkephalins, and the basic ones, dynorphins. 10 years after the cloning of opioid peptide genes, the receptor genes were identified. And these genes encode mu, delta, and kappa opiate receptors that were previously defined by the pharmacology. And it took another 20 years until enough of the recombinant proteins could be produced to be able to crystallize the protein and obtain the atomic structure of these receptors by X-ray crystallography. 2012 for the free receptor structures under an inactive state, and 3 years later, the first structure of the active mu-opioid receptor. I should add that the peptides, let's say, the selectivity for peptides to receptors is fairly low, although we know that beta endorphin and enkephalin preferably bind to mu and delta, and dynorphin preferably binds to kappa. I will not talk about the peptides. I will focus on the receptor, but we should not forget that the peptides are as important, although their biology is much more difficult to study. So let's talk about the receptors. 3 receptors, a distinct distribution in the brain seen here by autoradiography in the mouse, and a distinct distribution in the human brain seen here by in situ hybridization. And you can see from there that the distribution is very distinct for the 3 receptors. If you want to think of a general role for the opioid system, you can consider the system as a central regulator of basic processes for cell and survival. But this is very general, but basically this system teaches us what is good for us and helps us coping with challenges. And it does this by regulating reward and aversion processing, and this will be the focus of this presentation. But the system also clearly controls pain and nociceptive messages and helps coping with stress. And there's another entire research area about autonomies of peripheral functions of these receptors. So the outline of the talk will have 2 little parts, which will be, I would say, reviews, where our lab has contributed quite a bit, but many other labs also. And the 2 other parts will be focused on the mu-opioid receptor, on the circuits fulfilling the role of this receptor. And this will be much more experimental and will include unpublished data. So please bear with me until the end. Let's talk about this burning question. Well, it's burning since a long time, actually. Can we develop non-addictive analgesics? And maybe an important contribution was to show that if you delete the mu-opioid receptor gene, and this was 25 years ago, basically morphine has no effect anymore. Morphine is not analgesic. There's no morphine reward. There's no morphine dependence. And all other things that we consider side effects also were abolished. Conversely, morphine effects are intact if you use mice lacking delta or kappa receptors. So the very simple first take-home message is that this mu opioid receptor is, well, the molecular target for both therapeutic and adverse morphine effect. And it was clearly shown later that this mu receptor is the protein that explains the effect of generally prescribed opiates and abused opioid drugs. So given that the mu receptors is mediating all these effects, can we kill the pain without addiction by developing mu opioid agonists? And the answer is maybe yes. In fact, opioid receptor signaling is pretty complex. Activating any of the three opioid receptors will basically recruit G proteins intracellularly and other effectors. And this in turn will lead to short-term, almost immediate effects like decreased cyclic AMP and modification of several ion channels that altogether will lead to inhibit neuronal activity. So if I activate an opioid receptor, whichever I consider, I inhibit the neuron, either by modified hyperpolarizing the neurons or by inhibiting neurotransmitter release. And also there's other signaling cascades that lead to modified gene transcription, like for most G protein coupled receptors. Now, what is interesting is that the receptor alone does not explain everything. And in fact, what is important is the agonist receptor effector complex, and effectors are multiple. You can see here as an example, a survey of all the effectors that have been recognized or characterized for the mu opioid receptors ex vivo or in vivo. And you can see that there are multiple. Mainly some are G protein dependent and some G protein independent. So this leads to the concept of bias signaling. And in fact, in the GPCR world, we know that different drugs acting at the same receptor can signal differently. And why is this? Because the receptor, the active receptor, can adopt different conformations, and these different conformations depend on the drug and will basically favor the activation of distinct effectors. So you can have different effects in the cell with different drug acting at the same receptor. And this is what led people, so there's an enormous literature about that, to look at what we could do with the mu opioid receptor. And the mu opioid receptor is mainly bound, as I say, to GI pathways or beta-arrestin. And Laura Bone showed a while ago that if you lead beta-arrestin in mice, you have a better therapeutic profile. So this led many academics and companies to develop GI bias compound like the Trevena compound, which was developed using classical screening methods and which is now in clinical phase 3. There's the SR compound that was developed the same way, a very different structure. And there's the PZDM21 compound. This one is interesting because it was developed from actually virtual docking on the crystal structure of the receptor. And it provided a structure that is totally different from the known morphines. So we have a kind of new biology with this bias drug. But I must say that in the field, there's still this question of whether drug bias indeed explain the different therapeutic windows of each individual drug, because mu agonists have differential effect. And there's another theory that says that a low intrinsic activity or efficacy could also be the reason why you have less side effects and a better therapeutic window. So if you look at these papers there, you will see the current discussion, and it is probably something in between or both. It is probably that both the bias, so the ability to activate specific signaling downstream pathway, and low efficacy that will allow to develop a better drug. And this is simply to summarize all the efforts that are being done to develop new mu agonists with better, safer profile. Here in the middle, you have the prescribed opiates and the endogenous opiate peptide. I talked about bias agonists, but there's also other ways to do this. There's now very recently allosteric modulators have been developed that actually modulate the activity of the receptor responding to an endogenous peptide. There's also peripherally limited opioids and an interesting fentanyl derivative that doesn't cross the blood-brain barriers and only acts at inflammatory sites. And there's also ligand working at or acting at different receptors. So the take home here is simply that there's many strategies to reduce more mediated adverse effect. And also, of course, a more drastic one is to avoid the mu receptor. So now let me go to the second point, and I'll be pretty brief. So we have these three receptors. Let me focus again on the mu opioid receptor. I told you that if we delete the mu receptor, we have no more morphine reward. But also, if we delete the mu receptor gene, alcohol, cannabis, and nicotine are not rewarding anymore. They have no reinforcing effects. So it seems that the mu receptor is a key player, a common player for the rewarding effects of most drugs of abuse. I don't count cocaine here. Cocaine is a little bit different. Further, it seems that this receptor is also key for the mediation of natural rewards. And we know that these mice show less motivation to eat, and that these mice, when mice are pups, show reduced maternal attachment, so that social reward in these mice is reduced. And in adult mice, we also were able to show recently that these mice show some signs of autistic-like behavior. So the conclusion of the take-home here is that these mu receptors are important to mediate all types of rewards, artificial rewards, natural rewards. So if I summarize, here is the mu receptor. Activating the mu receptor is pleasurable. So now I summarize studies performed by many labs. Activating kappa receptors, on the contrary, is dysphoric. And these opposing activities of the two receptors on what we call hedonic homeostasis, euphoria, dysphoria, stems from their opposing effect on modulating dopamine signaling. Now, when kappa receptors, when the under exposure to stress or to chronic drug, the animals develop, let me start this differently. It was also shown that the kappa receptor activity or the kappa receptor dynorphin system activity increases under stress or under chronic exposure to drugs of abuse, and that this leads to depressive-like behaviors. So the activity of this receptor system is clearly on the side of the negative affect. On the contrary, the delta receptor there has anxiolytic and antidepressant property. So it is known now in drug development programs that if you want to control mood and improve mood, you should rather activate the delta receptor and inhibit the kappa receptor. So I don't think we should separate, this is a little bit artificial from my end, to separate hedonic homeostasis and mood control, because they are of course interrelated and circuits are overlapping, but I did this for, let's say, convenience. So now, this was the review, let's put it that way. Let's move to the next part of the talk. And first, let's do a little break about methods and approaches. For many years, we have been deleting genes and looking at the consequences of this gene deletion on behavior, but clearly we had no clue on the circuit basis of this behavior. To enter the circuits and analyze what happens in the brain, it is clear we can adopt two approaches. One that I call microscale level, so we can manipulate a gene or a neuron population at a targeted brain circuit, and then look at the consequences on behavior. And to this, we developed conditional knockout mice, knock-in mice, and also we have an entire armada of CRE mice to develop optogenetic and chemogenetic approaches to manipulate neurons. There's another approach, which I called mesoscale level. And this is now the exact opposite approach. We want to see the whole brain. So we could manipulate a gene or a neuron or treat an animal and see what is the consequence on whole brain activity. And this is something we approach using mouse Fmri. So our lab has been working well. We have been putting efforts in developing mouse fMRI since about 10 years. This was very, very, let's say, it was the beginning 10 years ago, but this has become now much more sophisticated. And I will show you data using the two approaches. So I will move from one to another. So let's talk about the Neopet receptor under physiological situations. So now we talk about models with naive animals, there's no drug on board, and we look at reward and aversion processing. Before we knock out receptors or we manipulate neurons in the brain to study a receptor function, we need to know whether receptors are expressed. And to know whether receptors are expressed is a true challenge because we know that autoradiographic studies have no cellular resolution, and we know that antibodies are rarely specific in vivo, particularly on brain section. We have to be careful with that. So we develop new tools. So these are genetic tools where the receptor is fused to a C-terminal fluorescent protein. And this is a knock-in animal. The first one was done by Greg Scherer in my lab. He developed the DOR EGFP knock-in mouse, which in fact expresses a fluorescent receptor in place of the native receptor. And the receptor is fully functional, so it doesn't alter the physiology. And you can see the receptor at macroscopic level in different layers and even at subcellular level. And you can even see the receptor molecules trafficking and internalizing in the cell under exposure to deltorphin 2 here. This approach proved extremely useful because it allowed us to revise anatomy of the receptor. It allows us to propose different paths towards tolerance based on receptor trafficking, and it allowed us to show that physiological internalization is very different from drug-induced internalization. So we were very encouraged by this success, I would say, and we decided to develop mice that express a fluorescent neuroreceptors. We have two of those lines, one which expresses a functional red receptors, mice that express functional yellow receptor. And you can see some of these beautiful images. And also we develop now in the area of GPR receptor other tools. We have mice expressing visible GPR88 receptor, chemokine receptor, neurotensin receptor. These tools are pretty wonderful. And using these tools, we can now study and identify exactly where are the receptor expressed, meaning in which cell type. So if we use these tools to map receptor expression, we will find that the mu receptor is broadly expressed throughout reward aversion brain centers. So what I call reward aversion brain centers or brain centers, that I show here, here are some of the many reviews that will summarize for you what I show. This is a very, very primitive scheme, and I apologize for that, but it's helpful for the presentation. We have more expression at all these sites. It is more or less important, high, medium, or low. And in yellow are what is typically the dopaminergic system is there. And in blue are sites of, I would say, negative affect. So we're going to talk about this. And first, I will summarize what is best known is the mechanism whereby neuroreceptors facilitate reward. And this is now has been demonstrated by hundreds of papers. We know that Neopet receptors are expressed on GABAergic interneurons in the VTA, and that when you activate a neuroreceptor there, you will inhibit those inhibitory neurons and enhance dopamine release. So this is a disinhibition hypothesis. Today, things look a little bit more complicated because we see and we know that there's these neuroreceptors are expressed also in other cells in the VTA, but I won't spend too much time on that. We have been able to delete the neuroreceptors in the striatum conditionally, and we have been able to show together also with William Wu Yang and the UCLA Opioid Research Center, that the receptors there at the level of the nucleus accumbens are also not only involved in drug reward, but in the motivation to obtain the drug. And I would like to mention that we have beautiful expression of the receptors in large cell of the lateral hypothalamus, for example, and septum, that are known as hedonic hotspots, and that there's still studies to do to find sites of mu-opioid receptor mediated reward. So this is the reward part, and the take home is that it is clear and obvious that more facilitate drug seeking and taking in dopaminergic circuit. Now, these receptors, as I said, are expressed here, for example, in the habenula, in the dorsal Raphid nucleus, in the amygdala, and in the bed nucleus of the stria terminalis. So we are used to think about the mu receptor, about the receptor generating euphoria, but what is the role of the receptor in those areas which are notoriously known to mediate aversive states? Just a comment here, I don't talk about the cortex here. I apologize for that, but there's no room for cortical in this particular talk, photocortical receptors. So the question was, is more activity influencing both reward processing and aversion processing? Where is this receptor acting exactly? So this is where we use fMRI, and I want to show you our first study. And a little introduction would be to say that fMRI is probably the best approach in human research to see the brain at work and to correlate genotype to phenotype, to find biomarkers for disease, and to study maybe drug effects, although this is more complicated. What if we would use and study this in mice? In fact, in mice, what would be interesting is that we would have not only images of receptor functioning in a live animal, but we would also be able to do mechanistic research, because now we can manipulate genes and neurons at will. Just a little message. There's a scale issue. This is a human brain. This is a mouse brain. So it's very, very small. So we need a big machine. And our first study was done with Laura Arsan, working in Jürgen Hennig lab in Freiburg, and our student. What Laura had developed is the measure of resting state. So just to say in a few words, what you measure is the bold signal, which is a proxy of neuronal activity in the brain. There's no stimulus. The mice is slightly anesthetized, and you measure slow bold signal fluctuation for each voxel of the brain. This is the resolution. The brain overall has about 100,000 voxels, so it gives you a little idea on the resolution. And what we measure are the spontaneous fluctuation of the bold signal for each voxel, and our statistical analysis will allow to study the synchrony among these voxels. That's the principle of the experiment. So the idea was to acquire data and simply compare mice lacking mu receptors, so constitutive knockout, and controls, and compare the connectivity patterns. So first of all, we look at voxels that are synchronous or oscillate together, and this is a total open-ended approach. Voxels that oscillate together are shown here, and what you see are components, and what happens is that these components actually cover meaningful anatomical regions. So these voxels define anatomical regions that works in a synchronous activity. Now what we can do is take each component and study long distance functional connectivity, which means we want to know if one component and another show synchronous activity. Are they correlated? Do they show synchronous activity? We can do the correlation matrices for control animals and for knockout animals, and we find something like this, which is called a correlation matrix, where each colored little square we show significant difference in the correlation between a pair of components, with the difference between wild type and knockout. So what we get here is a signature of more activity on the whole brain. Now, you may say that changes are everywhere throughout the brain. So we can apply other statistical analysis to rank the number of changes. And let me move to the next. This shows the components that are most changes. So this means whose connectivity has been most perturbated or most difference between wild type and knockout mice. And what you see here is that the most, so there's many more, it goes to 100. I show you the top 10. The most changes are actually brain area that process information with negative valence. You can find periapical gray, which is a pain center, the amygdala, as you know, a fear center, anterior cingulate cortex, amygdala, the habenula here, and the medial Raphid nucleus. So we decide, and note please here that the accumbens is very, very, very low, much below in the ranking. So it is clear that the neuroreceptor activity has a strong influence in shaping the networks with negative valence. Let me now go focus on the habenula. We chose to work on the habenula because this is an area where the neuroreceptor is most densely expressed, as you can see here. So the next question we had is, do neuroreceptors play any physiological role in the habenula? So where first, the habenula is now well recognized as an aversion center or anti-reward center, and has been shown involved in, let's say, in humans, in monkeys, in rodents, to be important for addiction, anxiety, and mood. And actually, manipulation of the habenula in humans, there are some cases, are able to reduce, let's say, depression. Only case studies for now. Where is the neuroreceptor? The neuroreceptor is expressed in what is called the diencephalic conduction system. So you need to know that the medial habenula is a tiny structure, highly conserved across vertebrates, and has two subdivisions, the lateral habenula, which is very much studied actually now, and the medial habenula, which is much less studied. And this medial habenula receives projection for the median septum and goes mainly to the IPN. And here you see, using more cherry mice, the neuroreceptor expressed in the medial habenula. You can see that the neuroreceptor is expressed upstream in these beautiful neurons of the septum, and is also expressed downstream in the main projection area. And because these more cherry mice or more Venus mice allow us to do this, we can do now co-localization study, and we can see that these receptors are expressed in the two main populations of the medial habenula, which are cholinergic neuron and substance P neurons. So what we've done is we have deleted the neuroreceptor specifically and only in the medial habenula. In fact, we have used a Cre mice that express Cre under the control of the beta-4 subunit of the nicotinic receptor, which is expressed preferentially in the medial habenula. And the result was that we got mice which lack mu receptors in 50% of the medial habenula neurons. All of the receptors are preserved. We tested mice in all the tests, in many tested behavioral tests. What we found is the following. You know that naloxone is aversive. If you use naloxone, you block the endogenous opioid receptor tone, and you're going to produce aversion. We all know that naloxone is the antidote to rescue individuals under overdose, because it will displace the drug very rapidly. And naloxone is also blocking the endogenous neuroreceptor activity. So you see, for example, here, a conditioned place aversion experiment when a mouse, a normal mouse, receives naloxone, and you see that the animal avoids the compartment. Now, in the beta-4 mutant mice, it seems that naloxone is much less aversive. This was a striking effect. So what we did is we repeated the experiment, and this is not published yet. So this is again a naloxone CPA conditioning experiment. In black, you have the control mice. You see that when we test the mice at day 8, 11, and 16, we have a strong aversion. And if we extinguish the conditioning, mice still avoid the compartment, and they will avoid this compartment until day 73. We didn't test later on. And in blue, you see that the beta-4 more mice clearly show less aversion. So we confirm that naloxone aversion is lower. And they actually recover pretty fast and get to full extinction. So what is clear is that the aversive effects of naloxone are partly, sorry, I made a mistake here, are partly explained by the activity, the blockade of neuroreceptors in the habenula. So the hypothesis here is that we have neurons in the medial habenula that encode aversive states. We have a neuroreceptor there, and when endogenous opioid come and activate this receptor, we inhibit this neuron and we limit the aversive medial habenula activity. So if the receptors indeed have an anti-aversion activity at this brain site, then the next thing would be to test whether these neurons expressing the neuroreceptor indeed encode aversive states. So this is a very recent study we have just finalized. First of all, if you want to manipulate neurons expressing neuroreceptor, you need mice expressing recombinase under the control of the Neopet receptor promoter. It took us a long time, about 10 years, to finally succeed in doing these mice. Why would these mice be interesting? First, to understand the circuit basis of endogenous opioid receptor activity, as we're going to do here. And also, of course, in the future, it will be extremely interesting to identify and study the neurons that are normally stimulated by opiate drugs and adapt to opiate drugs. So we created these cray mice. These are knock-in mice that actually we have inserted T2A peptide signal and fused to EGFP3 so that these mice expressed a large precursor protein encoding for the receptor and the recombinase, which is further cleaved in the cell to generate separately the receptor and the recombinase. And we were able to show that this receptor is fully functional and that the is functional. And we were also able to show that this is suitable for tracing study. For example, this is a kind of reconstruction we did where we have crossed the Morphine mice with DT tomato reporter mice. And now you can see each individual neuron that expresses the mu-opioid receptor. And we were also able to demonstrate that our mice are suitable for optogenetics. We have performed an experiment where we have expressed the dependent channelrhodopsin in the VTA in Morphine mice and found, using real-time place testing, that the animal expressing the channelrhodopsin indeed avoid the compartment where the stimulation chamber, which is what we would expect if we activate GABAergic neurons that actually inhibit dopamine release. So these mice are suitable. So now we're going to use these mice and try to optostimulate medial habenula neurons that express the mu receptor. So we inject the predependent channelrhodopsin here in the medial habenula of Morphine mice. This is the injection site. We place the probe above the medial habenula, so we activate everyone. And what you see here in a real-time place testing setup, sorry, it's real-time place testing, that the control mice show no preference. The channelrhodopsin mice show actually avoidance. They avoid the stimulation chamber. So we produce an avoidance behavior. This is simply to show that our optogenic stimulation conditions indeed increase CFOS activity only in channelrhodopsin mice, both in the medial habenula and IPN. And we're doing presently some electrophysiological experiments to characterize the optogenic effects on these neurons. Now, these neurons, I told you that the main projection site was the interpeduncular nucleus. We performed tracing studies using the Morphine mice. We injected in the Morphine mice a dependent reporter virus in the medial habenula. This is the injection site. We found, of course, a lot of fluorescence in the IPN. Interestingly, there's other sites. So, for example, one site is the Raphid nucleus, dorsal, ventral, medial. There are obviously projection, direct projection on the dorsal, on the Raphid nucleus. So now we decided to optostimulate either the medial habenula IPN pathway or the medial habenula DRN, dorsal Raphid nucleus pathway. If we stimulate the medial habenula IPN pathway, so injection in the habenula, the light above the IPN, we do real-time testing experiment. We find that channelorhodopsin mice show a strong frequency-dependent aversion. So this is obviously aversive. There's no change in the locomotor activity. If we do the same experiment now by placing the light above the dorsal raphe, there's no avoidance, not at all. Next, we started to test other responses, and in particular, levels of anxiety in the elevated plus maze, the open field and marble burring. And what you see is that stimulation of this neuron does not modify the behavior in the elevated plus maze, does not modify the behavior in the open field, not in the marble burring. But if we use a tail suspension test, which is a proxy of despair-like behavior, we have a significant difference so that the animals are more immobile. If we do the same thing on the medial habenula DRN pathway, this is the interesting thing. We get the exact mirror image, so that we have clearly higher anxiety in the anxiety test, and we have no effect in the tail suspension test. So this leads us to this idea that now, sorry, I need to, yeah. This leads us to the hypothesis that we have two types of neurons expressing the neuroreceptor originating from the medial habenula. Okay, I'm stuck.
So if you press on the arrow.
Yeah, fine. These neurons encode, let's say, negative affect or aversive states in different type of behaviors that we measure. Clearly, we get avoidance and despair-like behavior for the IPN projection and higher anxiety in the DRN projection. And then the idea is that endogenous opioids, or perhaps exogenous opioid, probably two, by activating the receptor would inhibit these responses. So this is interesting because it seems to further support the notion that these neuroreceptors may limit several aspects of negative affect through distinct circuit mechanisms. So the novelty here is that the neuroreceptor is mainly known as a pro-reward receptor, but we never thought about the neuroreceptor as an anti-aversion receptor at other sites. Of course, findings lead to next questions. Next question in terms of hedonic homeostasis will be, which are the endogenous opioid peptides here that act to reduce or refrain the aversive medial habenula activity? And the other thing is, what is the contribution of this mechanism to withdrawal and the negative affect of withdrawal? One hypothesis would be to test that the receptors become tolerant, for example, and that the tone doesn't operate anymore in dependent animal, leading to increased negative mode. Just to finish on the habenula, just to say that actually habenula research in the mouse is relevant to human research because we know that the neuroreceptor is strongly expressed in the human brain. This is an experiment we made using human brains from the Douglas Brain Bank. And you can see that mu, this is mu delta, ORL and kappa receptor, mu expression is very high in the habenula. And also we obtained Fmri images, resisting images from the Imagen cohort. And we were able to first to run independent component analysis experiment, which, as I explained you earlier, which are able to identify a habenula component. So there are synchronous voxels that we can identify that actually overlay the habenula area, although this is very, very tiny, 3 millimeters by 3 millimeters in the human brain. And we were able to show that in individual with the what is called the more frequent mu polymorphism, the connectivity of the habenula is different, with the rest of the brain is different. So this is work in progress. Now, let me go to addiction. So I will be very brief on the introduction because you all know this three-stage model where recreational drug use will lead to binge intoxication and then leading to a withdrawal state, which is aversive. Note that I always have reward in yellow and aversion in blue, blue like the blues. And then in this state, we then go to a preoccupation anticipation stage or craving stage, and we go back in the vicious circle. While the brain is more and more exposed to, for example, chronic opiates and the neuroreceptor is overstimulated, adaptations occur leading to lower reward, to dependence, to lower mood, and also to all these processes that are heavily studied in the field that I don't touch on today, which are basically the loss of self-control and the maladaptive habits that develop. So just as a kind of summary of the literature, we know that neuroreceptors are involved in the binge intoxication state. We know that the kappa and delta receptors are involved in this withdrawal state. And we know that all three receptors are one way or another involved in the more cognitive aspect of the pathology. Now, I'd like to draw your attention on the fact that exiting the vicious circle is very difficult. And in fact, maintaining protracted abstinence, which means long-term abstinence in the clinic, is known to be extremely difficult as low mood, irritability, and enhanced emotional responses increases with time, precipitating relapse. Unfortunately, if you look at the literature in basic research, the neurobiology of long-term abstinence is very little studied, and in particular, the neurobiology of the negative affect in the state of protracted abstinence. We developed a model, it's now 10 years ago, a very simple model where of protracted morphine abstinence. The animals are exposed to morphine, so it's a treatment, it's not voluntary consumption. However, these animals are heavily intoxicated, they become heavily dependent, and now we wait. We put the animals back in their cage, they go through spontaneous behavior, and after 1 or 4 weeks, we measure their emotional responses. And what we found is that after 4 weeks, only 4 weeks, not after 1 week, we have increased immobility in the tail suspension test, and we have reduced social interaction. So it seems there that an incubation of despair behavior and social interaction deficit that rises with time. We also found that we measured dopamine, serotonin, and norepinephrine in the brain, and we found that serotonin turnover remained increased after 4 weeks. And we also then, of course, tested fluoxetine effect, and we found that if fluoxetine is applied during the 4-week abstinence, we now reverse or we prevent the development of social interaction deficits. We also showed that this is also observed upon chronic heroin. The dosage is slightly different, but we find again a decrease in the social behaviors, and this lasts up to 7 weeks. It's very long lasting. We also find the despair behavior, and we also find working memory deficit. And in the case of heroin also, we found that fluoxetine is able to reverse this behavior. Finally, we found that in 4 weeks, we can actually prevent the development of this phenotype or treat, in a way, this phenotype, either by 4 weeks fluoxetine during abstinence or after abstinence, or using a kappa antagonist NorBNI. And what we find here is animals that have seen NorBNI during abstinence do not develop the social interaction deficit, and animals that have seen NorBNI after the abstinence phenotype has developed also reverse. So, driven by the notion that there's a serotonin mechanism involved, we actually deleted the neuroreceptors beforehand. So we used flux mice, we injected a Cre virus, we verified after 4 weeks that we had deleted receptor. In fact, we had deleted 40% of the receptors in the dorsal Raphid animal, then undergo the heroin exposure, and then we test, well, we measure naloxone withdrawal after 1 week on a separate cohort. So the withdrawal score is the same. So whether you have neuroreceptors in the Raphid or not, you get a somatic withdrawal. But for animals that have seen the neuroreceptors diminished, we clearly do not see the development of social interaction deficits, while the working memory deficits are the same. So it is interesting to see that, in fact, the chronic activation of neuroreceptor in the DRN is absolutely essential for the development of the social deficits. So now the next question is, if we skip microscale, what is the role of neopioid receptors and the role of neurons expressing those receptors in the Raphid nucleus? And this, in fact, has not been studied. And how do these neurons contribute to the negative effect of the abstinent brain? And we have started looking at neuroreceptor expression in the Raphid nucleus, and it seems it's not going to be a very easy task because neuroreceptors, in fact, in the Raphid nucleus are expressed equally in excitatory cell and inhibitory cells, and a little bit, but very little, in serotoninergic cells. So this is undergoing. And next, and this is the last part of my talk, let's go now mesoscale. And the question is the dorsal Raphid nucleus the most important brain region affected by the by the chronic morphine exposure? Is this the most important region? Is there a signature for abstinence? How important are adaptations in reward and aversion center brain-wide? So this is now the Fmri comes back into play. We now repeat this experimental design. Animals are exposed to increasing doses of morphine, get very dependent, go through abstinence, spontaneous abstinence, or withdrawal in their cage. We image the animal after 6 weeks, and we measure their behaviors after 8 weeks. And you see that after 8 weeks, we still can measure the increased immobility in the tail suspension test. So there's still signs of despair-like behavior. Fmri, how do we do this? A slight anesthesia. We set the animal in the scanner. We measure resting state for 10 minutes. We inject morphine, and we keep on measuring the bold signal. So what can I say? We take actually repetition time or 1.5 seconds. So we scan the whole brain every 1.5 seconds. The resolution is the same as I told you earlier. This experiment was done at the Douglas Imaging Center at McGill, and we have 108,000 voxels. We have completely revised and re-optimized all the pre-processing steps. So this is a very painful and long procedure. After we do all the pre-processing of the imaging, we get to the time course extraction, and it looks like this. And this is the type of signal we work with. For example, this is the signal a long time of the fluctuation of the bold signal in large left and right central amygdalas, for example, or in the retrospinal cortex, or in the anterior cingulate. So now we can do thousands of analyses. I will only show you selected data. We are only at the beginning of the analysis. We can either work totally open-ended, independent component analysis, no hypothesis. I will not show you this because this is undergoing. We can work seed voxel-wise, which is we sit on a seed that we have chosen, and we look how this seed correlates with all voxels of the brain. Or we can work on seed to seed. How do seed correlate? Let's look at the seed voxel-wise. I'm only talking about the basal state. So we don't talk about the morphine injection. We now only consider the 10 minutes of basal recording of the images. We have selected 23 seeds, which cover not only our classical areas of interest, but several cortical areas, 3 regions of the hippocampus, the basolateral amygdala, the central amygdala, caudate putamen, the pallidus, epithalamus includes the amygdala, several areas of the hypothalamus. We have the dorsal Raphid and medial Raphid nucleus, the VTA, periapical gray. We also have the parabrachial nucleus. We have a set of seeds, a set of, let's say, nuclei that contain the locus coeruleus, and we have also the respiration centers. And you can see these areas that we have chosen here in 3D in the brain. So we have now looked how voxels from one given seed talks to all the other voxels of the brain. And I show you the example of the Raphid. This is the smallest seed. It includes 119 voxels that you see here. And here, what you see are the voxels that are differentially correlated to the dorsal Raphid nucleus or to the DR seed in the abstinent animals. So the blue voxels show higher functional connectivity. There's none in this case. Yellow, red show lower functional connectivity in abstinent mice. And for the dorsal Raphid, what we find is that there's about 5,000 voxels throughout the brain that are differentially correlated to the voxels of the Raphid. If we do this for all 23 seeds, what we see is a ranking of seeds on the top that have the highest number of voxels modified throughout the brain, and those who have the lowest number. And you can see that this does not rely on the seed size, as the caudate putamen, which is the biggest seed, show almost no change in abstinent animals, where the VTA, which is very small, shows the highest number of seeds. It is nice to see if we look at the first top 10, which are truly strongly significant. There's a strong change in the connectivity pattern of those seeds. We have, first of all, the default mode network with retrosplenial cortex, which is the node of resting state network. And dysfunctional resting RSP means dysfunctional brain. Then we have learning, let's say, hippocampal areas or seeds that are changed here. In terms of reward centers, we have the VTA, globus pallidus, and accumbens. And we have most of the aversion centers here that are modified in the abstinent brain. You can see them here, the 10 top represented in space, and you can see them here. Now we can do another thing, and this is my last slide. We can work seed to seed. So we can see how each seed correlates with each other seed, and you can have a correlation matrices. These are correlation coefficient. Highly correlated pairs are in red. Very little correlated pairs, seed pairs are in dark blue. And this is a matrix for control. This is a matrix for abstinent animals. And they are different. And if you run statistical analysis, you will find a number of pairs that differ significantly between control and abstinent animals. And they are shown here. So what is shown here are the connections now, which significantly differ across between abstinent and control animals. It is interesting to see that we have lower functional connectivity between VTA and the dorsal Raphid nucleus, which is very much studied in the literature, but also, for example, between the anterior cingulate cortex and the lateral and the hypothalamus, a hypothalamic region. So, where do we go with this? First, we can do seed-to-seed analysis and cover the entire brain. We have developed an algorithm to cover 150 brain regions, which cover the whole brain. And we have this type of matrices. We can work, as I said, open-ended. We can work on the dynamic functional connectivity. I have no time to explain. We can try and work out predictive feature. Are there any predictive features from functional connectivity parameters that predict the abstinent brain? We can correlate with behavior. We also will analyze the abstinent brain under morphine, and we have another experiment at 4 weeks of abstinence. Why is this important? For basic neuroscience, I think that this will help us generate a novel testable hypothesis. And for medicine, and this is a very long-term goal, maybe we'll have translatable abstinence signature for diagnostic and treatment. This is usually what we say at the very end. We're certainly not there yet. And I will just summarize. I will not read the whole slide, just say that in terms of the mu receptor as a target, I think that organism level studies are very important in drug development, and there's a lack of in vivo studies to accompany drug development. In terms of physiology, as I said before, I think that the opioid peptide signaling is really understudied in vivo. In terms of neuroreceptors in circuit of reward and aversion, I think that the novelty here for us is that mu plays an anti-aversion role in the habenula, and the mu receptor, or the overactivation of mu receptor in the Raphid, clearly is important in the negative mood of abstinence. There's certainly many questions, but I won't go into that. And finally, regarding the Fmri, I think that this technology now is getting much better and is also key to progress. And I want to thank, well, the team we had for 6 years in Montreal, the new team now in my new institute in Strasbourg. I want to thank all the funding bodies, and in particular NIH, that has supported me all these years, and all my close collaborators listed here that have contributed to work I have shown you. And to end up, there is place and there is a package for an independent young investigator that would be interested in a related field in our new institute. Just contact me. And here I'm finished. I thank you, and I'm ready for questions. Sorry,
Okay.
I've been a little bit too long.
Thank you so much. Yes, we're already at the hour, but I'm sure we have a few minutes for questions.
Yeah.
So, Christina, there's already one in the Q&A.
Yes.
Please write
Can you
the
hear?
questions in the Q&A tab.
Yes. Okay. So someone is asking, Sergi Ferre's group has shown that neuroreceptors can form heteromers with galanin-1 receptors in the VTA, with galanin opposing the effects of opioids. To what extent do you think various heteromers modulate the effect of opioids and can be targeted for therapy?
Okay, I think I have a very quick answer. Heteromers have been demonstrated in vitro. Heteromers are also shown in some cases in vivo. It is very difficult to show. I am not sure that how much these heteromers are relevant for physiology. I don't know. My opinion is that remains to be demonstrated.
Okay, so next question, Christina.
Thank you. Yes. In which brain region did you find an increase in serotonin in morphine abstinent mice?
Yeah, this was in the dorsal Raphid nucleus, because we tested several brain regions, and this is where we found it. Of course, there's many projection areas, but there was nothing there. It was really happening at the level of the dorsal Raphid nucleus.
But those mice, were they showing depression-like symptom?
Yeah. This was done in
were.
mice. Yeah.
Okay, and another question here is neuroreceptor in habenula neurons to DR or neuroreceptor in DR. Yeah, dorsal rapid.
Does it play a role in depression?
Yes.
Well, we don't know, but what I've shown for now, we really progress step by step, is that it may be the case. Because if we activate, optostimulate these mu receptor expressing neurons, we produce a despair-like behavior.
So it might be,
now.
But still.
It might be, yeah.
Yeah.
And so if I may ask a question, so what is really interesting is that the high expression of these mu receptors in the medial habenula, and then next to this is the lateral habenula that is directly connected to the tail of the VTA, and a lot of work goes into it. And yet you prefer sort of the detour through the IPN. Isn't there a connection between the medial and the lateral habenula?
Okay, very honestly, I would prefer to see the mu receptors in the lateral habenula. But it's not the case, actually. They are located.
But you could have a system where you have the medial projecting to lateral.
Yeah. There's very little evidence for direct connection between medial and lateral. There's 1 or 2 papers in the literature, so it may be possible. The other difficulty is that most receptors or receptor expressing cells are in the medial, but there's a few in the lateral. So it's not, so at the moment, we do not know if the border between lateral and medial is a kind of strict border. Okay, so we're developing tracing experiments in the lateral and the medial in more mice to completely define where we are. And we're going to do electrophysiology there to see if there's functional connection between the two subdivisions.
Okay. So Christina, you have one more question or?
No,
Okay.
We're done with the questions. Yeah.
Good. Then let's stop here and thank again Brigitte for a stimulating talk, introducing a very important system and showing some of her latest results. Thank you so much. And for all the participants and of course also the panelists, please come back on October 7. We'll have Eric Nestler talk about epigenetics and in addiction. So thank you so much for being with us.
Thank you very much.
And have a
Bye-bye.
nice evening or day. Thank you. Bye-bye.
Bye.
Bye. Bye.